first webinar under our three-part series on biocontrol. The series is a collaboration between the ASEAN Action Plan on fall armyworm and CABI. My name is Alison. I'll be your moderator for today's webinar, and I'm joined by some wonderful speakers and also our technical support expert, Pranav. So you would have seen this hopefully just before. It's just a few quick pointers. Any technical issues, try logging off or on, or send a message to Grow Asia or Pranav in the chat box. You use the Q&A box to ask speakers to the questions. You can use chat just to give us a heads up about a point that you wish to share, or a link, or highlight something, or say thanks or great speech. Make sure, if you can, to rename yourself under the little tab there that says more. You can go in there and you can use perhaps your name and give your organization. So if you do want to talk or ask a question, we know exactly who you are. And 5 there is if you really have a burning question or really want to say something, you can put your hand up and we'll try to give you the chance to do so. It's not quite that I've just been told I haven't quite updated the proper names of the presentation, so don't look too hard at those titles. Most of them are right, but it's really just to give you an idea that we're moving at a pace today. We've got lots to share, and what we really want to do is use this as a catalyst for further discussion in the ASEAN Action Plan on fall armyworm, as a way of thinking about what could we be looking about going forward regarding projects on biocontrol. I've just got a quick poll here just to see who we are. We've got 347 people that have registered, which is fantastic. So Pranav, can you launch the poll? We've got 150 people with us so far. If you could choose, the host and panelists cannot vote. So if you're a panelist, I'm sorry, you'll have to just resist that urge to choose one of those options. I'm going to give people just maybe 5 or 6 seconds more. And then we'll get an early indication of who is with us on the call. Pranav? Oh, great. Oh, that's quite a spread. Yeah. We haven't got any farmers or growers, so we're going to have to work on that, I think, for next time, because that would be really great to see some people from that stakeholder group. But that's really, really good to see the spread there between government, research, private, and that's fantastic. It's pretty even, actually. Okay, so next slide. Let me see if I can do that. I don't know. I think the poll has to close for that. Let's have a look. Okay, I'd like to introduce now Graham Dixie, the Executive Director of Grow Asia, just to give a few thoughts and an introduction to today's session. Graham?
Thank you, Alison, and thank you for this incredible response. 347 people registering is amazing, and looking at the bottom there, I see the numbers rising up with every second. And I guess there's 2 reasons for that. One is the really impressive job that Alison did in drawing together such a community of interested and practitioners around this particular issue. And the other side is just what a big issue this is. And we did some rough calculations, and you know, if it hits Southeast Asia anything like as badly as it hit Africa, this could cause over $800 million worth of damage. And that's money taken out of the pockets of smallholder farmers. And it would be foreign exchange that would need to be expended from the region to import additional corn. What has been interesting about this whole piece is that this is the first time the ASEAN has actually thought about collective handling and tackling these regional issues. I had the privilege of having to present to the senior officials from the ministries of agriculture across all the regions, plus the Assistant Director General of the ASEAN Secretariat. And the point that came out from them was that they were very excited about this whole approach, partly because this was the first time that they were really converting something which the ministers had come up with into something that was now bottom-up development and was now actually rolling out. But they saw the potential in it, and they saw the potential that if we could prove that regionally we can work well together, if we can share information, if we can exchange new practices, best practices, if we can draw into our region global best practices, if we can make the research and the communication more cost effective, this would be a really big first for the ASEAN and for the region. And the second piece was that they said, if you can make this work, this would be really important going forward, because certainly the consultations that we've had across the sector is these kind of extreme events, whether they're pests or disease or climate, but in our case, pests and diseases, these are likely to happen more often. And the point they raised to us was, what would happen if the desert locust arrives here? How would we respond? And if we have a mechanism of a community of practice of doing the things that have been set out in that regionally developed bottom-up action plan, then we have a much better chance of handling this. So it's not only an exciting launch, but it is also something that we need to now prove that we can function together and that we can learn from one another, and we can come up with a modus operandi which works regionally. I think that's all I need to really say, Alison, but I look forward to this enormously.
Excellent, Graham, and thank you very much. And I'd just like to also thank not only CABI, who's done this in conjunction with Grow Asia and the ASEAN Secretariat as well. I'd like to thank. They're very important to this whole process, and all the members of ASEAN that have supported that. So a big shout out to those that have joined us from the ASEAN Working Group on Crops, and also the Expert Working Group on Phytosanitary Measures. It's a pleasure to have you join us. Now, Roger, you is now joining us. Roger is from CABI, who we've done this in collaboration with, so welcome, and I'm going to give you the floor and turn off my screen.
Thanks very much, Alison, and I'll just turn my video off now that you've seen me and know a little bit what I look like. So I'll turn off. Yes, thanks, Alison and Graham. We're very pleased to be co-hosting this series of webinars with you, which is looking at biological control. Next slide, please.
Oh, sorry.
Can I have the next slide?
Yes,
Biological control, I think most of us know what we're talking about, although different people refer to different aspects of biocontrol, but basically it's using living organisms to control our pest. And we talk about natural enemies, which may be predators, parasitoids, pathogens. In the case of an insect like fall armyworm, the natural enemies we're talking about are usually arthropods, other insects, or microbes, pathogens. And biocontrol is probably best looked at as the basis for integrated pest management, which everybody agrees is the approach, the overall approach we should be looking at. So in our webinars, we're going to be looking at the different types of biocontrol, and I've listed 4 there. So conservation is the first level, and that's basically encouraging the natural enemies that are already in the habitat through managing the habitat, also avoiding killing them. So things like spraying will kill the natural enemies as well as the pests. Augmentation is about releasing large numbers of natural enemies that we've reared specifically to release, to boost the populations that are already there. There's a category of biocontrol which some people say isn't properly biocontrol, but we're covering it here, and that's biopesticides. And these are products which are based usually on some kind of insect pathogen that we can spray like a pesticide, but which are based on living organisms. And then lastly, there's classical biocontrol, which we're going to be talking about more today, where we introduce a natural enemy that isn't already present. And often this is introduced from the area that the pest originates. Next slide, please. So in getting biocontrol used, I just want to highlight a number of areas we need to think about. As scientists, we tend to look first at the effectiveness. Does it work? And we say how much mortality a natural enemy might be causing, but we do need to go beyond mortality. We need to think about whether it's reducing the farmer's losses. But there are various other things other than just the question as to whether the natural enemy causes mortality. We need to look at the regulatory context. Some of these approaches have regulations governing them to make sure that we do it safely. So we need to think about that aspect, and we need to use international guidelines and standards where appropriate. We need to think about the practicalities too. Some biocontrol approaches might be a little bit difficult and impractical maybe for smallholders. And we also need to take into account gender considerations here. So social science is as important as the biological science. And lastly, we need to also think about economics. Are the approaches we're proposing cost effective? It's very easy to say we've got a natural enemy that kills lots of pests, but can we use it economically? Is it cost effective? So I hope we'll think about these different aspects as we go through our webinar series. And to get all these done requires collaboration. And we'll be hearing more about this as we go through our webinar series. But I would just highlight this research collaboration portal for fall armyworm, which you can look at later and perhaps participate in. Thank you, Alison.
Thank you, Roger, very much. And there were some very good thoughts there to take forward into the discussion after our presentations around the feasibility, effectiveness, gender issues, for example, to consider. I'm now going to introduce our first speaker today, Dr. Marc Kenis from CABI. Marc is an entomologist with over 30 years of experience in applied and environmental entomology, and he's currently leading the Risk Analysis Invasion Ecology section at CABI in Switzerland, and has been working on fall armyworm biocontrol over the last 3 years. Marc, you have the floor. I'll just move on to your first slide.
Okay, thank you, Alison, and hello everyone. I'm glad to see so many people connected. I will stop my video as well. You can already go to the next slide, Alison. So here we're going to talk about classical biological control. And as Roger said, so classical biocontrol is the introduction of a natural enemy of exotic origin to control a pest, usually also exotic, aiming at a permanent control of the pest. I'm not going to go through all the protocol because it takes too long, but this implies a few important things. First, it implies collaboration and work in the area of origin of the pest or in a country that has already introduced the natural enemy. You cannot do classical biocontrol in your own country without collaboration. It also implies introduction of exotic organisms, which involves, of course, some risks of non-target effects. Thus, risk assessments need to be made, including following regulation, national regulation, and including quarantine facilities and so on. But one of the biggest advantage of classical biological control, in contrast to the most other control method, is that there is no need for repeated actions, which means often huge benefits because the control is continuing year after year without further cost if the natural enemy is well established and works by itself. Next slide. Of course, I'm working more in Africa for the moment on the fall armyworm, and of course in Africa the main example of success in classical biological control against an agricultural pest is against cassava mealybug, another South American species. I'm not going to talk too much about this now because I know that in the following talk there will be more data on this immense success, because this had the following. The introduction in Africa dates from the 80s, but much later it was also introduced and successfully controlled in Southeast Asia. Next slide. But there are, of course, many other examples of success of classical biological control. We have counted that until 2010, we have a database at CABI, and have counted that about 700 insect pests have been targets of classical biocontrol projects worldwide, and 27% of the insects were controlled. This may look not that high, but this also includes projects that didn't finish or that were not carried out properly. If you take only the good programs, the successes is usually higher. Next slide. Of course, the next question then is, can these successes like the one on the cassava millibug be repeated for fall armyworm? Well, I will be totally honest here, and I have to say that it's a bit more difficult than other targets like the cassava millibug. First, fall armyworm is a pest in its area of origin. Many of these targets were not pests like the cassava millibug. So of course, by introducing a natural enemy from the region of introduction, you can expect success. Here, it's a bit more complicated because it's also a pest in its area of origin. Secondly, in America, fall armyworm natural enemies do not seem to play a major role. If you look into the literature, there are lots of papers mentioning low parasitism rate, for example, but I can say already now, I mean, I will be speaking a bit more about that later, but I can say already now that there are bias in the literature which probably underestimate the effect of natural enemies in the Americas. And finally, in Africa and Asia, fall armyworm is already attacked by a complex of parasitoids and other natural enemies, which is not always the case in classical biocontrol. And of course, you have to take this into account, not to introduce natural enemies that will compete too much with the already established natural enemies. Next slide, please. But I think it's really worth trying still, despite all these difficulties. Next slide. And the first reason, of course, is that fall armyworm is a huge pest with serious impacts on livelihoods, including human health. Nobody will contradict that. Next slide. And we can also argue that even a general decrease of population levels by 10, 20, 30% would have huge benefits because it is a huge pest which is widespread throughout 2 or 3 different continents. It means also that the total control is not necessary. Minor damage usually does not result in yield losses. So if you can lower the damage just by 10, 20%, you may reach a threshold where you have, for example, to diminish by half the amount of pesticide use, which across these different continents would be an immense success. Next slide. Also, population levels are definitely lower in the Americas. This is not always clear because it's also a pest there, but I've worked in both in Latin America and in Africa, and I can assure you that populations in smallholder fields in America without pesticides are less damaged than in Africa. I'm not so sure about Southeast Asia because I don't know the situation very well here. And next slide. And finally, we have to say that following classical biological control, some invasive pests have become less damaging in the area of introduction than in the area of origin. This is not uncommon. This has happened in several cases, and we may expect this also with fall armyworm. Who knows? Next slide. Of course, if we have to introduce a natural enemy from the Americas, we have to see which ones is or are the best. Fall armyworm has many natural enemies in America. If you look into the literature, there are lots of parasitoids lists, predators, pathogens, and so on. Predators we don't really consider in this program because the predators of fall armyworm are all highly polyphagous. Some predators of other pests can be very specific and can be used, but in our case, we have to focus on parasitoids, which in many cases are more specific than predators, at least on fall armyworm. Next slide. But we still have to see how we select the parasitoids. And there are, of course, different criteria because there is, if you look into the literature, there are at least 2 dozens of parasitoids listed as parasitoids of fall armyworm. And the first criterion, of course, is the impact in the area of origin. It's better to introduce a parasitoid that has already an impact in the Americas. But I have to say here that many literature data on parasitism in the Americas underestimate parasitism, as we can see with our ongoing studies, simply because parasitized larvae don't do much damage, they don't feed a lot, they pupate much earlier, which means that the damage is not visible and there is a strong bias in the surveys and sampling towards healthy larvae. Next slide. Another very important criterion is the specificity. You cannot introduce parasitoids which you will know, which are likely to have important non-target effects. But there are parasitoids in the literature at least that are known or thought to be specific to fall armyworm. There are 2 on the screens for the moment. And possibly there are also many other cases of sibling species or host-specific biotypes of apparently polyphagous parasitoids. So this needs to be further investigated, of course, in the lab, but also in the field, if possible, in the area of origin. Next slide. Another criterion which needs to be taken into account is the empty niches in the parasitoid complex in Africa and Asia. And there we have a very good example. The most abundant and frequent parasitoid of fall armyworm in the Americas is Schelonus insularis. It's a braconid, which of course would be the best candidate for introduce at first. But also in Africa and Asia, we have different species of Schelonus, which are also the main parasitoids of the recently introduced pest, which means that there may be some competitions going on between these 2 very similar species. Next slide. And finally, we also have to look at climate matching. For example, we may need different parasitoids or different biotypes of the same parasitoid species if we introduce in subtropical areas or in tropical areas, for example. Next slide. So what are we doing for the moment at CABI? Well, we have started surveys in Latin America a couple of years ago with local partners, with national partners, mainly in Bolivia and Nicaragua, also in Brazil, in Peru, in Ecuador and Colombia, and have found very interesting data, which I don't want to detail here, but may detail later in another talk. Next slide, please. And this has resulted in the importation into our quarantines in Switzerland of 2 parasitoids, which are presently the target of specificity study, biological and ecological studies. This is Schelonus insularis because it is the main parasitoids in most countries in Latin America, and Malafigmae, which is the most abundant, apparently specific parasitoid of fall armyworm. Schelonus insularis is not totally specific to fall armyworm. So we are focusing on these 2 species. I can already say that Malafigmae has already now been sent to our quarantine facilities in Pakistan for further studies, further screening tests on non-target species in South Asia, and it will be sent as well in Africa, in Kenya, and possibly Benin in the next months. Next slide. Yes, I just wanted to say that we had planned to introduce more, to import more species, but because of COVID, it's very difficult for the moment to ship parasitoids and to travel there and things like this. So that's why for the moment we are focusing on these 2 ones, but hopefully we can import 2 or 3 more species for further studies later on. So this is the last slide. What can we do in an ASEAN context? Well, of course, we can take advantage of the ongoing projects. This means that a classical biocontrol program in the region would not have to be started from scratch. We have already further data on the natural enemies in Latin America and on the specificity of some of the main parasitoids. So of course we can use these data to progress faster, but still there are still a few things that need to be investigated in Southeast Asia. The first thing, of course, is the native parasitoids that need to be known. Those already attacking fall armyworm, because this is a very important data. Before you introduce exotic parasitoids, you need to know what is the native natural enemy complex that already attacking fall armyworm. We also have to look at potential non-target effects on native species, which may be different from East Asia and Africa. Climate matching, as I said, is an important matter as well. And of course, we have to look at the regulations that are in place in the region. And with this, I have finished, and I thank you all for your attention and would welcome any question.
Great, thank you very much, Marc. If you could put your camera on, that would be appreciated.
Yes.
And I have a few questions. I'm just going to direct people. If you do have a question, could you please put it in the question and answer box? The chat is there. It's very useful as well, but it would help us if you put it in the Q&A. So with that, I have a few questions coming through, and I think you've sort of already answered this, but I'm going to answer it again. Ask it again. In Asia, we have fall armyworm in diverse environments. Do you suggest introduction of exotic natural enemies or search for native natural enemies in the ASEAN context?
Well, at first, before introducing exotic natural enemies, we need to know what is the native natural enemies that have adopted the invasive species. I think it's important because it also somehow has an influence on the selection of species, and this is also important for the follow-up studies after the introduction. So this is the first step to be done
Great. Thanks,
right
Marc.
now before the introduction.
And I think you had that in those steps. You sort of put out that as the first step, and then looking at potential non-target effects, climate matching, regulations and quarantine facilities, etc. How long does it take to sort of move through those steps? I mean, I know that there's no one answer there, but could you give me a bit of an indication of, are we looking at years of work, or could we do something quite rapidly, or what are your thoughts on that?
Well, of course, these are insects which develop quite rapidly in comparison to species that are, for example, univoltine and take a long time. So it can be fast, but it's a question of means of funding, and it's not always easy to get that kind of funding for research, simply because research is not so easy to be funded in this kind of program. But of course, yeah. To give an indication, we started our surveys for natural enemies in Latin America about 2 years ago. Now we are in the step of testing the species in the countries, and we can expect next year to release the first parasitoid if everything goes well. So it would have taken 3 years, but of course for Asian countries it can be much faster because we have already all the data on the main parasitoids now. So it can be in 1 or 2 years.
Excellent. You mentioned something further, sort of earlier in your presentation around the literature data on parasitism, underestimating that. How do you address that bias that you've noticed, or how do you think that could be addressed, that underestimation?
Well, by doing new assessments using different protocols. We take this into account in our African studies, and this is why we often have in African studies parasitism rates which are higher than those mentioned in the literature from the Americas. So this can be sometimes surprising, but I think it's also because we are doing things in a different ways. There are protocols to do that. I mean, yeah.
Excellent. Okay. I have a question here from Muni Rangaswamy, and it is, what do you think about Telenomus rumus? It was introduced from Asia to Americas for classical biological control of fall armyworm in the past. What do you think of the potential of using it here?
Well, Telenomus remus has never become an important parasitoid in the Americas occurring naturally. It can be used as augmentative biocontrol agent, but in the field it's a minor parasitoid. But it has in Africa, data for the moment show that it is a parasitoid. It is a frequent parasitoid of the fall armyworm. But Telenomus remus is not considered for classical biological control because it is present in all the regions where fall armyworm occur. It's present in Asia, in Africa, in the Americas. So it can be used as an augmentative biocontrol agent, but we don't consider it anymore as a classical biocontrol.
Excellent, and that's something that we may be able to talk about in our last session, webinar session in October. I have a question here that's sort of drawing together a few questions that have come through around different approaches. How important is it to use classical biocontrol, or to consider that in conjunction with other approaches, other biocontrol approaches?
Well, of course, classical biocontrol is perfectly suitable with other control approach. Of course, if you are using a broad spectrum pesticides, that's not really compatible, because you will also affect the introduced natural enemies. But of course, yes, it has to be part of an IPM approach. In some cases, in quite many cases, it was the only control method that was used at the end, because it fully controlled the pest. But we are not in that stage in the fall armyworm, and with fall armyworm, I don't think that it will be the case. We will always need an IPM approach, but if the introduction of a natural enemy, of an exotic natural enemy, can help, of course, this has to be really considered.
Excellent, Marc. Marc, there are quite a few questions that have come through. We need to move on to our next speaker. I'm not sure if you'll be able to see in the Q&A. You may, if you go into that, click onto that, you can actually see some of the written questions coming through, and we'll come back and answer those in the panel session as well. But if you do get the chance and you can see them, if you can write any replies, that would be most helpful.
Yes,
Thank you,
I can do. Yeah.
Thank you very much, Marc. And if you could turn your video off now,
yeah.
I will introduce the next speaker. And Charuwat Taekul, are you on the call right now? If you could just put your put your video on just quickly.
Yeah, I'm doing it right now.
So you can turn it off. You can turn it off for the presentation, but we can hear you and we can see you. So just tell me to move the slides when you're ready. And I'll just introduce you quickly. It is my pleasure to welcome you to the webinar. Charuwat has spent 8 years working at the Biological Control Research Group at the Department of Agriculture in Thailand before finishing his Master's and PhD, focusing on parasitoid wasps. And currently his focus is on parasitoids and grasshoppers for conservation and utilization. And he joins us today to talk about classical control in Thailand.
Hi, everyone. Can you hear me?
Yes,
yeah,
we can.
Okay, great. So today I'm going to talk about the classical control in Thailand and potential for biological control. And the outline of my talk, including our successful case and the challenge with fall armyworm biocontrol management. The first one is pink cassava mealybug, and then the next one is coconut leaf beetle and coconut blackhead caterpillar. Next slide, please. Next slide, please. As a government regulation, before we introduce natural enemies to the countries, certain studies need to be made as well as after they're imported. Before we introduce a natural enemy, the study of insect-plant interaction and the study of capacity to affect other native species. For example, the hyperparasitoid and the attack of beneficial insects, and are they likely to becoming pests? After we import, we also have a research on investigation of the contamination, specificity of the natural enemies, the study biology and behavior under quarantine environment. Next slide, please. So the first pest that I would like to talk is pink cassava mealybug. The outbreak has been reported in 2008. It's around 48,000 per hectares had been outbreak of pink cassava mealybug. And then the number reached higher in 2009, 230,000 hectares. So of course, insecticide could not keep it under control at that time. So we introduced Anagelas lopizai from the International Institute of Tropical Agriculture, Benin, in September 2009. At that time, we introduced 500 samples. We test for host specificity for the 6 beneficial insect and 8 pests. For mass rearing, we use mealybugs rear on cassava plant and later develop on pumpkin. Next slide, please. This is the picture of the one that we use conventional rearing on cassava method first. It's developed on pumpkin later because we found out that they can reduce cost of parasitoid production. As you see at the bottom of the slide, is the pumpkin and the cassava mealybug. All right, next slide, please. So for the activity that we done for field release, after having permit in July 2010, the recommendation we observed 300 pairs per hectare. We relieve 300 pairs per hectare. And then for heavy infestation occur, we release 1,250 pairs per hectare. The most important part is technology transfer. We have a training courses for rearing. So 44 parasitoid rearing unit established at the Department of Agriculture and Department of Agriculture Extension, Thailand Tapioca Institution at the cassava flour factory. And the successful case that we found, more than 15,600,000 pairs of parasitoid released during 2010 to March 2012. The outbreak area were reduced to 4,000 hectares and helping the cassava farmer and Thai tapioca industries. Next slide, please. So the next pest that I'm going to talk about is coconut leaf beetle. Coconut leaf beetle reported outbreak in 2004. The great impact to coconut plantation in Thailand. So at that time, we introduced Acicodes hispinarum from Vietnam in 2004. We got supported by FAO and Nong Lam University. The mass rearing of the host and the parasitoid were cultured in laboratory using coconut leaves. Next slide, please. So for the host, we use mature leaf for larvae. We use a young leaf for adults. And for Acicodes, the parasitoid, we have 80 to 100 larvae of the host and 50 adults of parasitoid kept in a plastic container for parasitization. Next slide, please. So as you see from the slide, after parasitization, the larvae of the coconut beetle are killed, turn to be black. And in the container, 20% honey solution smeared on tissue paper was provided as food of the parasitoid adult. And then when the mummy are collected and released after parasitization for 10 days, 90% released in the infested area, but we keep 10% for stock culture. Next slide, please. So for activity we have been done for field release, we're releasing 30 to 60 pupae per hectare for 3 times with 7 to 10 days interval depending on the damage. So as earlier, technology transfer is very important. We have been transferred to DOA Research and Development Center and Department of Agriculture Extension Insectary as well. After we release the parasitoid, they can establish 3 years later, coconut leaf beetle no longer become pest outbreak in Thailand. Next slide, please. So the last pest that we are going to talk about the successful case is coconut blackhead caterpillar or Opicinia orenocella. The outbreak has been reported, outbreak in 2007, around 11,000 hectares, approximately 5.56% of coconut plantation. Insecticide is not suitable for sustainable control. So we introduced Goniosus niphatidis, introduced from Sri Lanka in 2012. It has been considered as a potential parasitoid in India that we will research and introduce this guy to Thailand. So the mass rearing, we raise goniosas using both coconut blackhead caterpillar and rice moth caterpillar. And then after we do the research, we found out that the rice moth caterpillar is more efficiency to use as a rearing host. Next slide, please. This is the picture of we do research. We rear with both coconut blackhead caterpillar and we rear with the rice moth caterpillar. But right now, we only use the rice moth caterpillar. Next slide, please. This is the rearing process. We mating parasitoid for 4 days and we gently remove the female parasitoid into the container, having single rice moth caterpillar and keep container at room temperature. And then we maintain pupae parasitoid for 14 to 15 days before reaching an adult. And then mating adult for 4 days before releasing. All right, next slide, please. And the activity we have been done, we releasing 200 female per ride every 7 days continuously for a month. And we find out that the more releasing, the more having effective result that we are going to hear about augmentative later on. So for the technology transfer, technology has been transferred to Department of Agriculture Research and Development Office Center across the country and DOAE insectary as well. So the successful cases after goniosas established for 3 years later, coconut leaf beetle no longer become pest outbreak in Thailand. So next slide, please. Okay, we come to the challenge that we are going to talk today about the fall armyworm in Thailand. So since the outbreak of fall armyworm in Thailand around late December 2018, DOA generate the urgent control project to manage the pest, both chemical control and biological control. There are several experiments for the biological control. So we found out that the efficacy of sting bug and Bacillus thuringiensis or BT can control the fall armyworm effectively. So at the recommendation that we have is early install larva. We use BT at 80 grams per liter. And at sting bug, we release 500 adults per ride per time after 30 days planting. Next slide, please. Next slide, please. As a taxonomist, I heard about they attacked Thailand in 2018. So I asked the government for the classical control. I asked my director about import certain parasitoid from outside the country. And my director and all staff here recommend me to, we need to know what we have first as a native species. So as a parasitoid taxonomist, I went out at the field and spend like a year to collect the eggs at the field. And then I found that they have a certain parasitoid that we have. We have Telenomas, yeah, the black one, that I work together with Dr. Poleshek, Dr. Andrew Poleshek at British Museum. And then we found the white one, as you see from the picture, is a Trichogramma. The black one is Telenomas remus from morphology, but I need molecular evidence to support that T. remus or not. And then we also found Chilonas insularis, the Bacconid parasitoid that we have here in the field in Thailand. So my point, our challenge is how to rear them and how to rear mass production of them as much as we can and then release. So I think we have the one that we need to import already. Right now, we just need the process how to rear them. Next slide, please. I think I finished for my talk today regarding to the successful case and the challenge of fall armyworm in Thailand.
Thank you so much, Charuwat. That was an excellent presentation. I really liked all the pictures as well of how it all worked with the parasitoid. Great. We have lots of questions coming through. So I hope you're ready. Some of them might be actually answered in the panel session, so we may not get to everyone's question. I have a question here. Actually, how costly is it to do this work? I mean, is it extremely costly? Does it take a lot of resources? What's your views on that? If you would,
ask me again, please.
sorry, if you were to look at a specific project on developing this further, a particular parasitoid, how costly is it? Very expensive to do that work, or is it something that's quite cost effective? What are your views on that?
The challenge, the further research that we need to do is we need to find out the way to mass rearing the effective parasitoid, both Telenomus remus and Chilonas insularis. Yeah, and because of the specificity as well. So the cost is about the laboratory testing, and we still need a survey, more survey, because these two species that I found only the small area in central part of Thailand. We have yet broadened, keep our eye open to other area across the country. So the cost might be, I'm not sure, it might be not high and not low, something like that.
That's a good answer. Not high and not low. Sounds just right. I have a question here from Pierre Sylvie. Unfortunately, it seems that once the exotic species, the parasitoid, have been judged as successful, there are no more projects funded to follow the long-term effects. Is that so in Thailand, or in your opinion? What are your thoughts on that?
Do you think that about the FAW armyworm or the pest?
I think just in general, in that once there's been a successful project, do you follow up as a part of the research in looking at the long-term effects of any introduced species or an introduced?
Alison, you mean beside the FAW armyworm, right?
Exactly. So,
for the long-term one.
Yeah, once you introduce, I guess in general, once you introduce a new parasitoid to your country, for example, do you ensure? Do you follow up long-term potentially what is happening with it?
We are doing it right now. I mean, we have I mean, the Center of Department of Agriculture has 8 centers around across the country, and then we have the farmer and researcher in that area, and they submit the number to the government, to us, to the center, and then they also have the know-how. I mean, the mass rearing technique with them. So right now, I think everything keep it under control for these 3 pests. Yeah.
Okay, great, thank you. Now, here's another question from Fernandez Macale. He's saying there's been slow uptake or adoption of biocontrol, especially by many smallholder farmers in Africa, due to fear of non-target effects, or due to low awareness or slow action of the biological control agents. What do you think is the best approach in achieving adoption and scale with smallholder farmers and using biocontrol options?
This is a very good question. I talked with my friend or my staff and the director here how to use it in the small farm or the small farmer. If we have a big amount or a big population of pests, so the first thing they're thinking of the chemical control, right? And the FAW armyworm as well. For the high density, we use chemical first, but for the low pest population, we can use biocontrol to control it. So keep in mind that before using the chemical, we need to consider the IPM first, and then put the biocontrol as the main focus on that process. Try to reduce the chemical control, and then you will maintain sustainable system into your small farm.
Excellent. That's a very good ending for the session. We've got many questions. If you can jump on the Q&A, you may be able to see some of those questions, and feel free to type a few answers. But you will be joining us later on in the panel session, where we will get a chance to ask you all the questions that have not been asked so far. So thank you for a wonderful presentation. And I will move on to our next speaker, Dr. Rica Joy Flor from IRRI, who's going to be talking about exploring sustainable options for fall armyworm management in Cambodia. Rica, can you join us?
Yes. Alison, do you hear me?
Yes, I do, loud and clear.
Okay.
So you can start. Thank you very much.
Thank you, Alison, and good afternoon, everybody. We are indeed, this presentation is about a case of just exploring, coming from a country where biocontrol is not very common, in Cambodia. And next slide, please. And you can skip this one, yes. So let me preface this presentation with the why question. Coming from IRRI rice background, why is this relevant to us? In Cambodia, we are currently doing a project together with the IPM Innovation Lab and many government partners, looking at ecologically based pest management, which is developing a package that can harness insights from the ecology and behavior of different pests, and thereby helping farmers to reduce their reliance on pesticides. And with this project, we wanted to support innovations, particularly bringing out these alternatives to pesticides, sustainable ways to manage pests, especially biological control, which is very limited in Cambodia at present. And we are also looking into sustainable management of pest outbreaks. And so this presentation is exploring fall armyworm management with our partners, but as such, it is not classical biocontrol. Yeah, so next slide, please. When fall armyworm was expected to arrive in Southeast Asia, one of the initial activities that we did was to look into surveillance and looking at whether the pest is already present and what are the natural enemies that potentially may be existing. And after collection of some fall armyworm eggs, the General Directorate of Agriculture tried to culture these eggs and look at whether there are parasitoids that would emerge out of this. And they found some Telenomus species from this one. But we know that this is only the first step. Next slide, please. We are interested, and next, yes. We are interested in what are the currently available options in Cambodia. There are, at the moment, not many options for commercial biological control that the farmers can easily access with. And we know that farmers would often resort to pesticides. Next one. So we tried an experiment where we looked at first, what if the farmers did nothing as one treatment control. Also, we surveyed corn growers in an area that has seen some incidents of fall armyworm damage. And we asked the farmers, what is their current practice for managing the pest? And the most common one was just applying insecticides, specifically emamectin benzoate, and making it a double application in one season. So this was the most common. But we also looked at what does the pesticide seller, what do these companies, also from the literature that's being brought out to the farmers, what is being recommended out there? And the recommendation was to alternate pesticide. It's still majorly pesticide. It's chlorantraniliprole, and then followed by emamectin, and again, a third application of chlorantraniliprole. So we looked at then, in terms of commercial biocontrol that is available in Cambodia, perhaps we can test one, and that is Beauveria bassiana, is also available in Cambodia, mostly imported from India. And we had a treatment that is applying Beauveria 3 times in the season. Next slide, please. From this experiment, we found that biological control has high infestation compared with the pesticide options, but also a bit lower compared with doing nothing. And we also found, in terms of yield, one more please. Yes. In terms of yield, we found that the biocontrol application using Beauveria had a little bit higher yield than the 3 other treatments. So we are seeing that maybe it has potential, but we also wanted to see whether this results can be replicated. So we tested it again, and next slide, please. And the question this time was, oh, one more thing. We also looked at, of course, the farmers would be interested in looking at biological control if they can access it, and also whether it makes sense from an economic perspective to the farmers. And when we looked at production costs, biological control is expensive because it's not very common in Cambodia, mostly sold in the capital. It's not the normal pesticide that you can find from a village seller. So that makes the economics of it a little bit not so competitive to the pesticide options. But if you examine the net profit, it might be better than using 3 applications of pesticides per season. Next slide. We wanted to then test this again, but this time also examine whether a reduced number of biocontrol applications, thereby reducing cost, would be more effective. So we added, one more please, Alison. We added 1 treatment, which is biocontrol using, again, still Beauveria bassiana, but this time application 2 times per season. And next slide, please. This is still freshly harvested. We have not yet collected or summarized the yield data, but we are seeing similar trend in damage scores. And in terms of comparing it again, pesticides, much lower, doing nothing, much higher. But we're also seeing there's a bit of difference between whether you apply 2 times Beauveria bassiana or 3 times of the biocontrol, 3 applications per season in terms of the damage score over time. So what we're seeing is perhaps if Beauveria bassiana were to be used, it would have to be adjusted in terms of perhaps tweaking the applications, or maybe we will need to explore other types of biological control, and that's where we are at the moment. Next slide, please. We wanted to also survey other natural enemies, other parasitoids, perhaps predators that exist in the corn areas, also maybe other entomopathogenic fungi, and then try to test the effectiveness of these collected natural enemies. At the moment, the General Directorate of Agriculture is still continuing the mass rearing of Telenomus, and we're going to explore whether we can do a mass release of this fall armyworm parasitoid and examine the efficacy of this mass release. So that said, next slide, please. These are the different partners that are working on this with us, and my colleague, Ms. Liberty Almazan, is also here, and we would be happy to answer your questions.
Excellent. Thank you very much, Rica. That was a very interesting presentation, and it was nice. We've had 2 sort of focus presentations on classical biocontrol, and it was nice to sort of end with that almost an introduction to biocontrol of the process that you've gone through to sort of start thinking about what could be the approaches that could be used in Cambodia. So that was a very nice way to end. I know there's going to be lots of questions coming up on your presentation. So Liberty, have you joined us on the the speaker. If you could unmute yourself, because I think you might want to answer questions as
yes,
well.
I'm here. Yes, I'm here.
Welcome. So Liberty's going to join Rica for this Q&A session, and I'm just going to bring up the question and answers for myself. So sorry, the questions, so I can see them. And here they are now. Okay, so, we've got lots coming in. Firstly, I've got a lot of questions about farmers and how do we get farmers to think about biocontrol? How do we get them to consider using it? I guess it's you're actually the perfect people to sort of answer that question from your social science perspective. What are your thoughts on that, Rica?
Thank you so much. I think that there has to be several prongs with which we help the farmers to access or to be aware of biological control. The first one is on the policy side, where indeed we've been talking about regulation for classical biocontrol, but there's also regulation in terms of selling and trading biological control, which is very important in countries where they're still starting at a baseline. The products are not yet there. To help the market to develop these products and to make them available to farmers in a cheaper way, that sort of regulatory policy context has to be ready. We're also looking at, of course, the state of knowledge. To what extent is your extension system supporting these options, and how much knowledge is available or being brought out to the farmers in a way that farmers can access it? Lastly, also accessibility at the local level of these options. We're seeing, for example, in Cambodia, kind of competition where the farmers are easily able to access pesticide, easily able to access the recommendations from pesticide sellers, easily able to see that pesticide is the most convenient or maybe the better option for them. So then that really leaves out your biocontrol technologies on the side. So if perhaps you have several prongs of strategies that need to be addressed, and all these together help the farmers to access and be able to use it and see that it has benefits.
Excellent. Thank you very much. That's a great answer. And
Yeah,
I think,
can I add to that also?
yes, please.
Yeah, I think it's very important to create awareness in farmers that there are other options other than the pesticides, other than the insecticides. Okay, if we can let farmers know these are other options that would give us benefit in terms of non-target organisms, in terms of the health of the environment and the safety of the ones or humans using it, then I think farmers will be enlightened and try these other options.
Yes, thank you.
Yep.
Thank you for that. Now, I've got some specific questions here around why did you only use bassiana? Did you try any other isolate to know the efficacy? Thank you, is the question from one of our participants.
Okay, thanks for that. We used Beauveria bassiana because it was the only commercially available option in Cambodia that we know. If the research would show that this is a good option for farmers, we can immediately recommend it at a national level that this exists and you can buy it. So that's where we started with Beauveria bassiana.
Well, that sounds like that's a good place to start, given that it seems it's the only available option. So you didn't have too many options to choose from. But here's some more questions following that up. What particular stage of the plant did you apply the bassiana?
So the Beauveria bassiana was timed in three spaces. So the first one was 2 weeks after planting, and then every 2 weeks thereafter. So it wasn't so much, and I had a discussion with Dr. Buyung Hali, and he mentioned that perhaps one way to maybe see better if Beauveria would work is to adjust the timing, not so much timed in this stage, but perhaps to look at the cycle of the fall armyworm and target it better.
Excellent, thank you. When here's another question. When spraying of pesticides is keeping the fall armyworm levels lower, why do you think the yields are lower? Did you check?
I think for that one of the things that we discussed was perhaps although the damage on the leaf or on the plant, looking at control, looking at biocontrol option, those treatments, perhaps the damage on the plant was there, but the yield, the plant itself was able to recover from this. Because we do not see, also during that time, we did not see other problems on the corn itself compared to this season. For example, we saw fall smut, we saw stem borers. So, but for that previous season, we did not. So we think that perhaps during that season, the corn was able to recover more.
Okay, excellent. Okay, another question here. We've got some very specific questions. It's generated a lot of interest. What dose of Beauveria bassiana did you use in the experiment? There we go. We're really getting down to the specifics here.
Dose of Beauveria bassiana.
Yes.
Oh, yeah, yeah. I think we used 250 grams per. If we convert it, it would be 250 grams per hectare per application. Because we only then used, and in the end, it was more like 750 grams per hectare.
Okay, great. And if you want to add to that, I think you'll be able to see in the Q&A questions, you might be able to provide some further feedback, but there's obviously a lot of interest, Rica, on this. I've got a question here which is a little bit different. Have you surveyed whether there's any fall armyworm infestation on rice?
Yes, we are very keen to hear about this. I must say we did not do a field survey as such. We are also depending on our partners in the provinces who are aware of fall armyworm on corn, but there has been no report that there's any infestation or damage of fall armyworm on rice so far in our 4 provinces where we work. So hopefully it stays that way.
Yes, let's hope so. Here's a question from Cyril Tura. Is it possible to combine botanicals to control fall armyworm with parasitoids? Perhaps you can have a go at answering that, Rica or Liberty?
Maybe Liberty? Would Liberty have better?
I think you can combine because botanicals are from natural sources. So I think it would work hand in hand with the biological control agents.
Okay, thank you very much. Here's another question for for either of you. What rating scale did you use for the fall armyworm infestation scoring?
Yes, we follow the FAO scale, the one in the biocontrol guidelines of FAO.
Excellent. Okay, how can we? Oh, here's a question here. Other than plant damage, sorry, I'm trying to read the questions and ask them at the same time. Did you collect the insect cadavers to confirm the infection by the Beauveria bassiana?
You mean the cadavers of the fall armyworm?
Yes.
No, we did not collect this.
Okay. The next one was here, do the farmers accept this method in Cambodia? I think you alluded to this a little bit in your presentation. You definitely talked about the farmer uptake, but maybe a little bit more insight into how easy is it for farmers to actually apply or use this method?
We have tried in our other experiments for rice. We have tried working with farmers using Beauveria bassiana for rice. in some cases, particularly in areas where they were used to spraying insecticide, and then they realized that, oh, there are safe alternatives and I don't have to spray it so much, and now I'm seeing the yields are okay. After 2 seasons, the farmers were quite happy with this. Part of the biggest disincentive was just the cost and the accessibility of Beauveria.
Yeah. Excellent. Okay, thank you. And one more question here, and I think we'll then go into our panel session. But I have here a question from Srinivas. What was the trigger for spraying or applying treatments, or is it calendar based?
It is calendar based. Yeah, and perhaps that is also something that we might need to reflect on moving forward, whether there might be other triggers that are better. But because, for example, for the pesticide treatment, we surveyed it from the farmers or from the pesticide sellers, and they were also calendar based in that sense.
Oh, that's very interesting. Actually, I just have one more question I'm going to squeeze into your session. Here's the last question on the Q&As coming through. Do you have a sense on rice or maize farmers' perception on biopesticides in Cambodia? Do they find it safer or more efficacious?
My sense is at the very beginning, there's a lot of skepticism for things that are not being sprayed. But spraying in general as a skill, as a practice, is quite accepted. So if it's biopesticides, that spraying, there might be a bit of openness for it. But then when it comes then to looking at pesticide safety, there is a lot of farmers that really say, oh, we would like other options that are not pesticides. But again, they would then encounter cost issues, they would encounter issues about accessibility, credit from the pesticide sellers. These things the farmers balance among these decision points.
Excellent. Thank you very much. And thank you very much to both of you. If you can stay on, we're going to have a panel discussion now. We have lots of questions still unanswered, and some of them have been answered. But if anyone else has now burning questions to ask any one of our panelists today, please start putting those questions in the question and answer box. If you've got something to add and you want to say it verbally, we may be able to give you the chance to if you put your hand up. So please think about that as well if you're brave enough to take on the panel verbally. Good luck. But if Marc and Charuwat, could you join us, please?
Yeah.
Yes.
I am.
Yeah,
Excellent. We have all of you there. Thank you very much for coming back. As you can see, I see some people have already answered some questions. Marc, I think I saw you'd already addressed a few. So thank you for that. I've got a question for all of you, and perhaps we could start with Marc, and this is a pretty wide-ranging question here, so you've got the hard one to start with. What do you think is the potential for classical biocontrol methods in Southeast Asia over the next 5 years?
Well, yeah, as I said, I think it's worth trying because it is something that is sometimes difficult to predict the success. But once it is successful, that's the very good thing because you don't have very much to do after it. So yes, I think we should try. And also, as I explained, even partial success would be immensely useful here. If we just can reduce by 30% the use of insecticides just by classical biocontrol, that would be wonderful already.
Excellent. Thank you, Marc. I have a question here. Charuwat, actually, maybe I can ask you the same question. What do you think from your perspective of the work you have done so far and the success stories in Thailand with classical biocontrol? What do you think is the potential for the next 5 years on fall armyworm control using the classical approach?
Yeah, there are more than one the parasitoid that we introduced into our field. If there are several scenarios to take it into account. For example, when you introduce parasitoid into the field, we need to reduce the use of insecticide. We need to reduce the pesticide and put more biocontrol. For example, the Beauveria, the BG, the NPV, or other natural enemies. Try to, because the parasitoid is very sensitive to the chemical, right? So the successful case need to come together with the IPM.
Excellent, thank you.
Okay.
Rica, a question for you. There's lots of different organizations and people and experts that are working across Southeast Asia in labs or out in the environment and also internationally. How do we bring these people together in a more efficient way, do you think? Do you feel like you're connected to what's happening across Southeast Asia?
I must say that there is a tendency for researchers to work together because of their natural connections. They already know each other. I think where we are connected, for example, with the IPM Innovation Lab, we get to know other colleagues in Nepal and Bangladesh also working on this. So then we find out about their work. So it goes with like a natural connection, personal connections of the different researchers. And I think that's always the fallback situation. But I have the feeling that where there's, for example, this kind of more specific topics, classical biocontrol, biopesticide, things like this, then there's a lot more interest specifically to engage with other scientists and to get to know what they're doing. And I think that's a starting point.
Thanks, Rica. Charuwat, what do you...
I agree with her. Yeah.
Yeah. Because I know that there's a lot of other interest in a lot of the Southeast Asian countries. I know Vietnam is doing a lot of work and is very interested as well. And I'm sure there is, just looking at the questions that are coming through on this chat forum and the question and answers section, I mean, there's people from across Southeast Asia here asking all sorts of detailed questions. The interest is there. How do we harness that more, do you think?
How do we what?
How do we amplify that interest more and get people working together on this? Are you connected, for example, into other laboratories and work across Southeast Asia?
I mean, currently we have a connection. I know people from Cambodia, from Vietnam. We have, yeah, like Dr. Rica said about, we have the initiate starting point, the good starting point right now, that we know people in Southeast Asia right now. We know the center. The next step, as we do in Thailand, the successful case in Thailand, we try to do it in the center in Bangkok of Thailand, and then we transfer the knowledge across the country through the DOA center across the country. And we do the same thing. If each country in Southeast Asia have the pilot project or the pilot grade project, they can share and distribute and help a whole continent to get success together.
Yes, that's a good idea, and I think that's something we could work on in the ASEAN Action Plan, is to bring some of those pilots together to share what is happening, and make sure that does happen, which I'm sure it will. I've got a specific question here around, it's actually back to you, Rica. It's around, does calendar spraying increase cost?
I must say that in Cambodia, the perspectives that I'm hearing is that where it seems like the pesticide companies are saying, oh, calendar spraying will keep the farmers to this number of sprays, to this correct pesticides. But if you look at also the practice of farmers, they are also quite efficient, really considering their own costs. So as much as possible, as much as they can, they also want to limit the costs. So I think where the farmers are oriented with IPM, they are looking, are able to look more into the natural enemies and base their pesticide spraying on that knowledge. But where there are farmers that are really depending on the pesticide sellers, their costs are much higher because the pesticide seller is not only telling them calendar spraying, it's also mixed in every application of pesticide.
Excellent, thank you, Rica. A question, Charuwat, from Singapore here. Are the parasitoids commercially available, or are they developed internally by the Department of Agriculture and distributed to farmers for use?
I mean, for other pests beside fall,
Yes.
beside these three pests, we working on some parasitoid, but it's not quite have a great impact or successful yet. Yeah, we have some, and right now we introduce the spider mite from Netherlands. Yeah, but we are working on it. We not yet get the successful. Yeah.
Thank you. And Marc, there was a question before that has come up again. It's this question of how much research happens after you introduce a species. How much research is sort of ongoing into the future? So it may be very successful, but are there checks to make sure that there's knowledge around what is actually happening going forward?
Do you mean after the success, or? Yeah,
so you introduce a parasite, predator, or pathogen, and is there ongoing checks to sort of see what is happening, even though it could be very successful, just to ensure that you have got that host specificity, I guess, in the long term, and not any leakage to non-target species. What are your thoughts on that?
Yeah, this is a major problem, is that usually donors, either they are happy because it worked very well and they want to spend money on something else, or they are not happy because it didn't work and they don't want to spend more money on something that first failed. So there are lots of biocontrol projects that end up with the release without follow-up releases, and this is a major issue in all continents with classical biocontrol. And yes, no, that's usually these studies are taken over by more academic grants at national level.
Okay, so that's an important consideration, I guess, in the design of any release in classical biocontrol, is those long-term work that goes with it. I've got another question here, and I'm not sure who would like to answer this. Maybe I'll throw it out there. We'll see who answers first. We've collected some parasitoid from the egg mass of fall armyworm. Where could we send that to have molecular identification? Marc.
Well, if it's on fall armyworm, we are welcoming any parasitoids for molecular identification for the moment. I'm not sure how long it can last, but yes, for the moment we can try to identify them. Yes, so that's possible. Of course, there are countries where it is not easy to get parasitoids or other insects out of the countries because of national regulations. But of course, it depends on the country.
Okay, so we know who that person is. So what we might do is, it sounds like, Marc, you're offering to maybe not do anything specific, but at least we'll pass on your details, and perhaps they can have that conversation with you offline as well. So we can do that as well if anyone's got any specific questions. Now, I've got a question for Rica. There's a lot of questions here around your Beauveria, In this case, maybe the host may be affected by other natural enemies. How do you estimate or know that it's from the Beauveria bassiana? Rica.
I must say that we did not measure the other parameters of other natural enemies or other potential things that might compete with the effect of Beauveria in the first season. In the current season, we collected some parameters for just documenting what are the other insects that are present as they are collecting the damage data. But I haven't yet seen this full set of data in the second season. So I'm not sure how to answer this.
Excellent. Well, thank you for giving that a go, Rica. I've got a question here for Charuwat. Is the DOA Centre doing some work on entomopathogenic fungi to control fall armyworm in Thailand?
You mean the using Beauveria or the other fungi to control
Yes.
fall armyworm? We are doing it right now, but last year, but the effectiveness is not quite as good as a BT, Bacillus thuringiensis. Yeah.
Okay, great. Thank you for the answer. Marc, I have a question for you. Although you published a very interesting article on Telenomus remus, I have noticed that you didn't support using egg parasitoid and classical control in your talk today. Could you just give a brief response to that? And your microphone is off, Marc, so I'll just get you to turn it on.
Oh yes, I was switched off because I was answering this particular question and I didn't want to hear.
Oh really?
Yes.
Sorry.
Okay. No. So I will answer orally. Well, indeed, but of course this is because Telenomus remus is already present in Africa and Asia, so there is no need for introduction as classical biocontrol agent. It can be studied and potentially be used as augmentative biocontrol through mass and regular releases. But for the moment in Latin America, when we are going in, when we are surveying egg parasitoids, we cannot find any specific or almost specific egg parasitoids in Latin America. So we are not considering egg parasitoids for introduction as classical biocontrol agent for the moment.
Okay, thank you, Marc, and thank you. I must have been reading your mind, seeing that you were answering that question. I'm going to end with a question to Charuwat and Rica. Firstly, just to congratulate all the panelists, this person says, for your excellent presentations, but they would like to ask a broader question. What are the 2 things you would recommend we should do as part of the ASEAN Fall Armyworm Action Plan in enabling biocontrol as a major component of the control strategy? So what would be the 2 big things you would recommend we should do as part of the ASEAN Fall Armyworm strategy? Who would like to start? Rica, I'm going to put you in the hot seat there.
Okay, I can think of one right now.
One is okay.
Yeah, I think that at the moment, at the top level, even at the level of the government, the regulators, the researchers of the national systems, in many countries, they are still not oriented with biocontrol and what it can do, how much, what is the effect, what are the potential risks. And I think that is an area where we first target, because these are the people that will continue the research and also bring out extension messages later on. So I think that's an area that we should also prioritize.
I think that's a great answer, and just that one in itself is totally enough, I think, for a start there. Charuwat, do you have anything to add to that as a 1 or 2, just briefly, you think are the key things we should be working on?
The key thing is we come to the right track. We need to know each other. We need connection. I read the paper of Marc Kenis, but I never see him in person. Yeah, but right today I see him in person. So next step, I may write a letter to him talking about what other effective parasitoids to control for armyworm beside Telenomus remus. Yeah, so I know Rica today. Yeah, that is the point. It's a good starting point to get connection in this region. and know each other and share the information and help each other. Yeah.
Excellent. Well, that's another brilliant answer there. Marc, I'll give you sort of what from a sort of looking into the region and being an expert in this area of classical biocontrol, what would be your one thing that you think we should focus on? You can have two if you'd like to, but they have to be brief.
Well, I'm not a specialist of Southeast Asia, so I'm not going to talk about farmers and strategies and connections and so on, but just on a scientific point of view, both for classical, augmentative, and conservation biocontrol, we need to have a very good data set of the natural enemies attacking fall armyworm presently in the different ASEAN countries. So that's the first step for all biocontrol strategies in terms of science. Of course, I know there are other issues in terms of social science, in terms of politics and so on, but in terms of pure science, I think that's a first step.
Great, thank you very much. And thank you to all three of you today. I know it's been very fast, and thank you to everyone else. We've gone slightly over time, but I do want to just move to a quick summary from Roger Day. He's going to be very brief here, and I'm sorry, Roger, for cutting your time off, but I think those summaries from our speakers there actually said a lot about where we could be looking for our next steps on this work in classical and biocontrol in general. Roger, can you just join us quickly?
Thank you. Yes, very, very interesting talks and obviously a lot of interest. I think we peaked at over 200 participants, which is fantastic, and lots of very pertinent questions. So there's clearly a lot of interest here. We need to find ways of continuing this discussion. And I think that's the main take home, I think, from the discussions, the importance of collaboration, which Charuwat emphasized, and indeed the other speakers have. We need to collaborate at different levels between scientists in different countries, different regions, more so when we get to some of the other approaches between the private sector and the public sector. We need to collaborate between the scientists and the regulators, between the social and the natural scientists. So there's various dimensions to collaboration which are critical for success in all these approaches. Classical biocontrol clearly has a lot of potential. We shouldn't look at it as the only possible approach, so all these other methods should be pursued. But classical biocontrol clearly has potential, and we've seen that there's capacity in the region, so we should be building on the expertise that's already in ASEAN, as well as the work they've already started. So I see this as an excellent starting point, and I look forward to the further events and collaborations that can be organized under the ASEAN plan. Thank you.
Thank you so much, Roger. And I'm just going to come back. I've got my friend behind me. As you can see, there's his face. Look, it's been a wonderful session. I really congratulate everyone for presenting, but also participating and asking lots of interesting questions. We will try and come back to you with some answers to those questions in the next few days, and you will get a copy of the presentation. Just note that we do have a webinar next week on technology potential for drones and digital IPM, which will be interesting, and we have those other biocontrol sessions as well. So please join us. I'd just like to say thank you very much, and thank you from ASEAN, CABI and Grow Asia, and MAD Vietnam, who is also the task force chair of the ASEAN Action Plan. And we wish you a very good evening or very good morning, depending on where you've joined us from. So thank you very much. And thank you, Marc, Charuwat, Rica, and Roger, of course.
Thank you very much.
Okay, bye. Thank you very much.
Bye.
Bye. Bye. Thank you.
Thank you.